In this introductory video, we introduce section 1.1 of your book and begin with the definition of biochemistry. A definition which is commonly used, particularly for dealing with lay people such as grandparents, is the chemistry of life. The problem with this definition is that living things are composed of lifeless molecules. Another problem with it is that it does not clearly define what chemistry is, and that what chemistry is fundamental to understanding the biochemistry. Therefore, a better definition of biochemistry is the study of the structure, property, and changes of biomatter. The way that I've got this course structured is that the first portion of the course is going to consist of the study of the structure and properties of molecules, and later on we will get to the more interesting part of the course, which is the changes of biomatter, where we talk about metabolism, such as glycolysis and the Krebs cycle. The science of biochemistry dates back to a time far before we actually understood that what we were doing was biochemistry. Your book has a very nice picture of an Egyptian pictograph denoting the production of wine. This is a form of biochemistry known as anaerobic glycolysis or fermentation. I would not really focus on things as ancient as this as being the origin of biochemistry, though. Instead, I would move us closer to the modern times, and there are some other things your book talks about that you should read, one of which is the synthesis of urea. It turns out that it used to be thought that the molecules of life could not be made by anything other than life forms. Then, quite some time ago, it was discovered that we could in fact synthesize a molecule of urea, which is, as the name implies, excreted in urine. This emphasizes the point that I made earlier, which is that living things are composed of lifeless molecules. And so I am going to talk a lot more about the origins of biochemistry in terms of the golden age of biochemistry, beginning with enzymology in the 1920s and going through the incorporation of the use of radiotracers around the time of World War II. And this brings us to another topic that we will introduce here and bring up periodically and regularly throughout the course. That is, what are some of the tools of biochemistry? The use of radiotracers, which I just alluded to, is really something that helped move biochemistry from a field in its infancy to the most powerful field of science for a period of time. Radio-labeled molecules, again, lifeless molecules with lifeless radioactivity incorporated into them, allow us to trace the pathway of metabolism and follow those molecules through various tissues of the body, as well as look at different molecular forms. Some other tools that I will introduce here, just as a list, and add more to later on, are things such as centrifugation, chromatography, electrophoresis, as these are some of the workhorse tools of biochemistry. A third point that we need to introduce here is that biochemistry is an interdisciplinary science. By definition, biochemistry is both biology and chemistry. And as we go through the course, what you're going to find very quickly in Chapter 2 is that we are using physics and basic chemistry to describe the interaction of molecules. So biochemistry is clearly an interdisciplinary science.